The development of a fertilised egg, a single cell, into a whole human being seems to defy belief. It's like some kind of biological miracle. But that egg contains all the information needed to make a baby that will grow into a child and on into an adult. Over the years, scientists have uncovered many of the secrets of that incredible transformation of a single cell into a whole living organism. Cells divide countless times to make tissues that grow and fuse, fold and dissolve in an intricate but reliable way to make a myriad of different living organisms. But development isn't just about cells multiplying. They also need to change into different cell types. This process of differentiation produces skin cells, bone cells, the cells in your liver and your spleen, the cells which form the crystal clear lens in your eye, and the neurons in your brain. In organisms that reproduce sexually, information comes from the joining of male and female gametes. Whether the organism is a plant, an insect, a human, or a zebrafish, development of the fertilised egg produces a miniature version of something about midway between its mother and its father. Understanding this incredible process of growth and patterning isn't just fascinating in its own right, it is hugely relevant to human health and modern medicine. So join us on a journey of discovery into the fascinating world of developmental biology.
So at its core, developmental biology investigates how genes, cells, and the signals they use to talk to each other generate an organism's heterogeneous size, shape, and structural features. This all happens in the astounding but highly reliable process of development from embryo to adult. It tells us about the differences between species and the similarities between individuals, or even twins. Some 200 years ago, Charles Darwin wrote about how embryonic development may be the key to understanding the similarity and divergence between species. From that simple statement, we have gone on to understand that developmental biology involves a complex set of processes including cell division and migration, patterning and morphogenesis, and cell differentiation and regeneration to give us the many cell types that make up our tissues and organs. Those discoveries, the ongoing research and the continuing advances have had a tremendous impact on human health, leading to many life-changing treatments. But for now, our journey must start at the beginning, with the essential formation of shape, known as morphogenesis. Hi Helen.
Oh, hi Courtney.
How are you doing?
I'm good. Welcome to the lab.
What are you working on today?
I can show you. Why don't you grab a chair? We can sit down and we can have a chat. These are just some adult fruit flies that we're preparing to set up for next.
So Helen, I'm a PhD student also working on fruit flies, Drosophila. Can you tell me about this word morphogenesis?
Yes, so morphogenesis is a really fundamental process that happens in embryos and actually within tissues within the embryo that really helps them develop their shape as an embryo develops. Now, the fruit fly Drosophila has been really instrumental in helping us understand how these morphogenetic events happen, and that's because the fly embryo, just like a human embryo, undergoes many morphogenetic events such as dorsal closure and gastrulation, which really help us understand generally how different morphogenetic events in us happen. I have a model of an adult Drosophila here, and you can see if we take this section of the body off, you can see inside the brain, the gut, the airways, the heart, many of the same tissues and organs that are within us. So dorsal closure in the fly involves the migration of two epithelial sheets over the surface of the embryo, and those two epithelial sheets move across the surface to seal a hole in the embryo, and they have to zipper up to close and seal that hole. And that's a very similar process to wound repair. And ultimately what we want to do is use the insights from our work in the fly to develop new therapeutic approaches to help us accelerate tissue repair in the clinic.
seat. So we work on how the embryo develops, and we work at the earliest stages when very fundamental differences are set up, such as which end of the embryo is going to be the head end and which end it's going to be the tail end. And how the heart, which is the earliest organ to form, how that emerges and how it's shaped so that it can do its job properly. It's fascinating that this forms from something that doesn't look anything like a heart to begin with.
Yeah.
It's just that crescent of cells in mammals, and even in fish, it's more like a disc, right?
Yeah. So the heart starts as a disc, which then forms this linear tube, and then from there, that linear tube is going to undergo this really complex bending and looping morphogenesis, which then gives rise to the heart. So my lab is really interested in understanding how you take this sort of seemingly simple heart tube and you turn it into a really morphologically complex organ. And we're particularly interested in how the environment around the cells influences that process. So within the heart, for example, there might be different extracellular environments made up by this thing called the extracellular matrix that are different in different regions of this simple heart tube, that allow that simple heart tube to fine-tune the different shapes that it's going to make as it's undergoing morphogenesis. I love that I can go to work in the morning, I can put some embryos under a microscope, and I can really see all of these processes happening in real time.
I wasn't always interested in how the heart forms. What really got me into how shape emerges is plants. Especially as a school kid, learning about the different shaped leaves, different types of flowers, different types of branches. I was really fascinated by how life forms can take so many different shapes.
So Tom, animals undergo these complex morphogenetic processes. Is this similar in plants?
It's very similar in a sort of conceptual way, yes. So morphogenesis, as in animals, is critically important in plants to make functional tissues. There are lots of differences too. Plants are surrounded by these cellulose cell walls that are there to help them keep their shape and to stop them bursting. But that means that cells are connected to their neighbours for life. They can't move, they can't change the neighbouring cell. So cell migration doesn't really happen in plants. In animals, that's a key part of development. But in plants, they have to solve that problem of morphogenesis without being able to move the cells around.
And how can this knowledge be applied?
I think as time has gone by, I've got more and more interested in trying to understand how we can apply developmental biology to make contributions to food security. So if you consider something like a leaf, the bottom of the leaf is covered in small pores called stomata that are there to allow the exchange of gas and water with the atmosphere. And so if you can understand the mechanisms by which stomata are patterned within the leaf, then you can work out how to optimize the number of stomata to achieve that goal of more photosynthesis for less water. So actually there's a lot going on here from a developmental perspective in what looks like a simple structure.
We've learnt a lot about how plants, trees and crops develop by studying a simple plant called Arabidopsis. Similarly, much of what we've learnt about human development has come from work on so-called model organisms from the animal kingdom. With these models, we can study how early cell divisions progress into something with a recognisable head, tail, a beating heart, circulating blood and so on. And we can look at what goes wrong when the genetic programs that direct development are disrupted.
So we work with the mouse, which is a great model for mammalian development. Just like humans, it has a 4-chambered heart. So Emily, this is a human embryo, this is a mouse embryo, but they look so similar.
You're right, I mean, they both look incredibly similar. And, you know, this is why we use model organisms to be able to study human diseases that have developmental origins.
There is no one best model for any process. It's important to choose the correct model for your question.
When people have a heart attack, it causes damage to the cells in the heart, and humans aren't able to repair that damage. But zebrafish, on the other hand, are able to regenerate their hearts if they suffer an injury. So if we can try and understand how the zebrafish is able to do that, hopefully we can take those principles and use them to be able to improve regenerative capacity in humans.
It is mind-blowing. During development, things are very fluid and continuously changing shape. Things which are seemingly separate objects merge together because their component cells can allow that to occur and then form new shapes.
Developmental biology can show us how cells move, bend, and change their shape to build the early structures of life, with some travelling long distances in the embryo to reach their final destination.
Oh, Courtney, hi!
Perhaps the masters of this kind of migration are the neural crest cells. Hi Karen, can you tell us about this wonderful population of neural crest cells?
So neural crest cells are a really interesting population of cells. They arise in the embryo of vertebrates, so they form at the back of the head, and then during embryogenesis they crawl out, and then they have to crawl through the embryo really long distances to innervate their target tissues. So that is, they have to basically invade the organ systems that they're going to become part of. The neural crest cells are unique because they have the capacity to become all different kinds of cells, ranging from bone and cartilage in the head structures, to neurons throughout your body, to the pigment in your skin, as well as things like neuroendocrine tissues. When things go wrong with the neural crest, things can go very badly wrong, leading to congenital anomalies, but also to pathologies such as cancer. A lot of cancers have hallmarks of embryonic neural crest, so they're multipotent, they're migratory, and they travel long distances.
In our lab, we study how neural crest cells migrate. We use the zebrafish embryo because we can film cells as they move within the embryo, and we can watch all sorts of very dynamic behaviors that the cells do. As they squeeze through the tissue, they will change shape, the nucleus will change shape, and we can really look at these in real time. Certain types of cancer directly derive from neural crest cells, and for example, one of these is a neuroblastoma. Neuroblastoma is the second most common solid tumor in children. And what we think is that when neural crest cells migrate, by squeezing into small spaces, they can experience mechanical stress, and this can cause damage to the chromosomes. We think that this may contribute to generating errors in the genome that may cause eventually malignancy.
We cannot do this in a computer system. We cannot do this in a two-dimensional cell culture system either, because we really need to understand how these cells contribute to normal function in a normal system. It will be really informative for things like anatomical anomalies that we see in newborns, for understanding cancer cell behaviors, which are really basically pathological cell migration behaviors. It's amazing to look down a microscope and to see this intact system coming together. I get to look at these beautiful embryos. I get to see life developing. I get to try to put together the pieces of the puzzle. And I get to try to figure out what goes right, what goes wrong, and how all of that's put together.
These zebrafish embryos are minute. They are less than a millimetre across, but there are already organs here. I've seen the beating heart. I can see blocks of muscle tissue forming. All those organs are already starting to function. And this development is being driven by the information that's contained in the DNA that was there in that single fertilised egg to begin with. Biologists worked out that the information needed to build an organism was contained in chromosomes more than a century ago. Then the uncovering of the detailed structure of the DNA in chromosomes paved the way for developmental biologists to work out exactly how genes control development. And now that science, developmental biology, is driving huge advances both in biology and also in human health.
So this one is from around 24 weeks of pregnancy, and you'll see it's almost completely smooth, and just the first few primary folds that are forming. This is now at 30 weeks, so that's more similar.
When we consider something as complex as the human brain, it's amazing that its development happens so reliably. But it's not completely determined by genes. Environment also plays an important role. If we look at identical twins, who are natural clones sharing the same DNA, the detailed structure of their brains can look quite different. The process of developmental programming is remarkably consistent and precise in some areas, but allows for variation, personalisation in others.
So how does folding differ in our brains?
Yes, so your brains are actually very similar because you're identical twins. Across the whole population of humans, we all have the same primary folds. So these are the really big folds that you see on the brain. So if I give you this one and I pick up a younger brain here, these are the first folds that form, so these really large ones here, and those are really similar between every single person. But when we get down into the super fine folds, the tertiary folds, then they start to be unique between individuals. But because you guys are identical twins, the folding pattern in your brains are much more similar than they would be between your brain and my brain.
So why is it so important to study human brain development?
So this brain that I've got in my hand is a great example of just how wonderfully complex the human brain really is. But we understand so little about how it functions. But now with new MRI technologies and movement correction, we're able to really get a window and see how our brains are developing during pregnancy. And what we hope to be able to do later is then say, okay, well, if we know how the brain is built, when it starts to go wrong in disorders or degenerative conditions, we might be able to then help alleviate some of those symptoms. But to me, that's one of the most exciting things about working in developmental neuroscience, is there's so much that we don't know yet that we need to explore.
In our lab, we are very interested in brain evolution and evolutionary comparison, and also trying to understand what is so special about the human brain. So this is actually a model of a human brain, a developmental brain, and you can see that we have two hemispheres, and this is the cortex. And if we look inside of it, if we can open it through, you can see that there are actually cavities. These are filled with the fluid in our brain, and you can see the different layers that are colored differently. I work with cerebral organoids and in general organoids in the lab, and we use them to study and look at development and evolution. So an organoid is a sort of miniaturized version of an organ, although it doesn't really look exactly like an organ. We start from stem cells and we aggregate them, and they basically form and arrange to form parts of an organ and partially recapitulate its function.
So what would an organoid look like?
So yeah, I can actually show you. So we have a model of an organ, and this is obviously a much bigger model, but you can see if you look inside, we have the similar cavities also filled with fluid, and we have similar layers. So in the lab, we generate organoids that can recapitulate brain barriers. So this is also a powerful model to study potential therapeutics and investigate drug permeability in the brain.
So how big are these in real life?
So yeah, they're not this big, but we can grow them in these dishes. And in this, actually, this is a 6 centimeter dish. We have 6 growing in this dish floating around, and you know, they probably develop around 7 millimeter maximum. So another advantage of this technique is that we can generate organoids from individuals with certain mutations that would lead to neurodevelopmental disorders. So we can then study and generate these organoids and understand how the cells that are mutated in that sense behave in a 3D environment, and we can then test potential different treatments to reverse that. There's still a lot that we don't know, and there's a world now that is opening with questions because we have now a new tool to use to answer them.
So we have seen how some model organisms are better suited to ask particular questions, which is why some labs will use one species, whilst another lab might use another one. And together, they can tell us how, for example, a brain develops, or how a limb is built, or how something as complex and beautiful as an eye is formed. So Rod, can you tell me a little bit about eye development and what types of questions you ask in your lab?
So initially the eyes will develop from a single group of cells that come from the same region from where the brain develops. But this group of cells will bud laterally and form left and right eyes. One of the questions in my lab is understanding how, even though these cells or group of cells develop independently, they will still achieve a similar size. There's an intrinsic property in the eye vesicles that enable eyes to grow to a certain size and then stop growing. That's what we know so far. And we're doing a lot of research to understand which are the molecular mechanisms involved in this process.
So how does this progress further?
So later, the eyes will undergo extensive cell rearrangements and foldings to form the neural retina and the retina pigmented epithelia. The neural retina will have all the neurons that enable sight. The retina pigmented epithelia, or RPE, will give shape to the eye and provide the physiological cues to sustain the cells in the neural retina. The research we do in animal models is relevant to understand human eye development too. I love watching embryos developing. In a few hours you can see how cells move, how tissues form, how the heart starts beating, and that to me is something remarkable.
Developmental biology reveals an astonishing world within us. Each of us is the product of the developmental process that created an entire human body out of a single fertilised egg. But for all the excitement that comes with a scientific discovery, what creates real, tangible impact is the end application. A breakthrough treatment, a human story, like the recovery of someone's sight.
We're introducing a new paradigm for the treatment of diseases at the back of the eye. A new paradigm because what we're trying to do is rejuvenate a part of the eye where those cells do eventually stop working and die. In Doug's case, what he had was a very bad bleed at the back of the eye in which those cells started to die. And as a consequence, he was going blind. What we managed to do was surgically replace those lost cells with cells that we'd made from stem cells.
Everyone wanted to go outside when the rain finally stopped.
He couldn't see the book that we placed in front of him prior to the operation. Now he's reading nearly 50 words a minute. The crucial thing for the whole program was understanding how those cells are first born. How do they actually develop in our bodies? We call them RPE cells. It stands for a very long name, retinal pigment epithelium. We have to put 100,000 of those cells into a patient's eye. Now that sounds a huge number, but actually that just covers an area of 3 by 6 millimeters. And in fact, what I'm holding in my hand is actually a patch, literally a monolayer. It's a single cell carpet and it's on an underlay of polyester. And what I'll be doing today, I'll be going to the Roy Free Hospital to get one of these patches because we will be operating on a patient later this afternoon. So we're continuing the program, we're continuing to treat patients, and the goal is to ensure that we show that it's safe, efficacious, and we can do larger numbers of patients. Taking this whole program forward wouldn't have been achievable without having that developmental biology underpinning how we actually make a stem cell turn into an eye cell. It's actually life-changing for those individuals which are given a second chance for sight.
I hope we've been able to share some of the wonder of developmental biology with you. It's an incredible story, that origami-like transformation of a single cell into a whole organism. And this science is about a fundamental question, of course, one that all children at some point will ask. How are babies made? We're getting closer to answering that question, and it's a huge team effort.
It's really difficult to get your mind around it. There are lots of moving parts, and it is just so counterintuitive.
It's absolutely beautiful. It's incredible being able to watch this process as it happens in a living organism.
It is very easy to start with one question and then find another one and branch out and get lost.
There's so much we don't know, there's so much to learn, there's so many questions we could ask, not enough time in the world.
There are different communities that I tap into that I think make me a better researcher.
I moved here from the US over 20 years ago, and the developmental biology community here was so welcoming.
I work just down the corridor from a colleague who, when I was a PhD student, we were together at BSDB meetings, you know, winning poster prizes and probably tearing up a dance floor somewhere as well.
All the discussions about science that went on outside of the talks, over dinner, over drinks, it was great.
A diverse community is really important because you never know where your next insights might be coming from, and sometimes it can be really unexpected and surprising. But to me, that's also sort of the excitement and the beauty of biology as well.
Isn't this the most incredible science? Coming here to the lab, finding out about what's happening in the cutting edge of developmental biology, meeting the researchers, it makes me want to come back and do another PhD.
Hi, I'm Paul Martin. I'm President of the British Society for Developmental Biology.
Are you looking into the lens? Or
No, I'm looking at you. I can't keep my eyes off you. You're so handsome.
Hi Helen, it's nice to see you. What are you working on?
Oh, hi Courtney, how are you doing?
Sorry. It tells us the differences between species. That's wrong, let's go again. It was good, but she had the wrong hand and she knocked the chair. I knocked the chair, I walked into the chair, she ruined it, Courtney ruined it. Yeah.
It's getting less and less natural.
Action.
I don't have Paul here telling us we must. I didn't say the word origami at all.
It is really beautiful, this science. It's better than physics.
That's definitely going to get a lot of people ringing more fields.
In essentially this... Oh, it's annoying, isn't it?
But I'd say, Pete, not again.
Did you get that on camera?
It's a rat.
Boom!